Welcome to another video tutorial. In this video, I will use a new and updated version of iDEP. In the previous video, I have already shown that this tool iDEP is actually used for RNA-seq data analysis, and this tool is helpful in doing RNA-seq analysis without any coding program. Hey, in this video especially, I will deal with many questions which were posted on my previous video, and in this version many of problems and errors have been fixed by their programmers, and moreover many bugs and new feature have also been updated. So I will try to show you step by step so that you can do by yourself. So the first important step which everybody used to ask, that which file we need to upload here. If you didn't watch my previous video, it is recommended to watch the previous video first. So you can use two types of data to upload here. So the first one is the read count data, and the second one is FPKM values. So I can show you the data which I have received from the company. So actually I have received this data from the company. We can see that this is the counts data and this is FPKM annotation data. This was the mutant type versus wild type. So in the wild type is actually the control type, and this is my mutant. So this is all gene. I mean, this file usually contain all the gene, I mean significant and non-significant. So we know that in RNA-seq data analysis, this file like mutant versus wild type with the differential p value is less than 0.05 and FC value more than 2. So this file contains only the significant number of genes. So I would recommend you to use this file so that you can check only those genes which are significant. So let me show you how we can change the counts data into how we can use the counts annotation data, and how we can remove the useless data. So here I can show you which data we should keep and which data we should remove. So I will use this count annotation file to upload. I will remove and get my significant genes using my own criteria. So usually we need the first line, that is gene IDs. The next three are mutant type samples. Usually we know that each sample has 3 repeats, so 1, 2 and 3. These are the reads count annotation data, and this is the wild type 3. So all here is not needed, so you can just delete all this data. And now this file you have to save in the form of CSV. So you can just simply export this data. You can go here, and we know that here is a CSV file. You can just save a CSV file, and that CSV file you can upload to that iDEP website. So now let's go back and check what is added in this new feature. So first of all, it is important that you can select your species. If you don't know your species, you can leave this option as such, so it will automatically match with the best species. So but for because samples are rice, I can use in scientific name Reza sativa and indica group. So here I will upload read counts data. So I can show you I have already saved that CSV file. Here I have saved the CSV file. So we can see that in this CSV file, the first is gene ID. So it is also important to note that here gene IDs, most of the people have posted some questions that their ID is not recognized by the system. So it is recommended to use those IDs which are similar to the famous databases like NCBI, Phytosome or Ensemble. So don't use personal IDs. Sometimes the company use its own IDs of their database. So it is recommended to use IDs which are published in famous databases like NCBI, Ensemble, or Phytosome. So here are the first three repeats. So my mutant name is APA1331. So I have just renamed, so sample 1, 2 and 3, and similarly wild type 1, wild type 2 and wild type 3. So this file I will upload there. So go back, I have selected the Oryza sativa indica group, and now I will start to upload that file. So here is the counts annotation data which I will upload. So you will simply click on open. So it will start to upload your counts annotation file in the form of CSV. So we can see that now our file is uploaded, so we can start the pre-processing, which I have already demonstrated in the previous video. So but before that, I want to show you that in this feature, many of the features have been updated, like they have added a network program, and moreover many bugs have been fixed. So here I can show that the gene ID have been uploaded. So here we can see that this is the gene ID. This was our first sample, second sample, and third sample. Similarly wild 1, 2 and 3. So now we can switch to the pre-processing. So here is the pre-processing. So we know that it's actually the total read counts in millions. So we can minimum CPM, you don't need to change, you can just keep it as 0.5, and number of libraries equal to 1. So all these data you can keep it as such. So there were actually 32,582 genes in totally 6 samples. So among them we can see that 21,724 genes passed this filter, which we have applied this filter. So 1,317 were converted into ensemble gene IDs in our database. The remaining 8,000 genes were kept in using the original database. So now we can move ahead. If you want to save this figure in high quality form, so it is recommended that you can open this like high resolution figure. When you will click on this, so it will just open this file into Adobe Illustrator, from where you can save it in the form of high 600 or 300 dpi photos according to your need. So I can show you how we can save these files in the form of 300 or 600 dpi photos. So let's wait. Yeah, you can simply click 1. Yeah, from here you can some of my previous file already opened. So actually they say the text is not found, but no problem, we can choose a new one. Yeah, here is that file which we have just imported. It is also saying that, oh okay, no problem, we can change the text. So this is file which we imported from there. So you can go there, export, and export as. So you can upload in the form of TIF. So simply click on here, export, and here you can change your DPI from 300, medium, or 72. So let's suppose 300, if we want to save it on 300, so you can click on OK. So it has saved your file. So similarly, you can also download that list in the form of Excel, that is converted counts data. So if we click on this, we can see that this is a converted count. So that list from which we have the system has converted into ensemble gene IDs and can be used for further analysis. So you can keep this, or while submission of your manuscript you can submit along with this. So the next important step is the heat map. So here it is automatically generating a heat map of all all significantly related DEGs, although which will start from here, but we can also start make a heat map according to the genes expressed in our mutant type and wild type. So we can see that here mutant type 1, 2, 3, but here is 2, 1 and 3. So it has actually rearranged, so you can click on this. Do not reorder or the cluster sample. So when you will click on this, so it will change it into a organized form like 1, 2, 3, 1, 2, 3. It has been reordered. So I personally prefer to use this color instead of green, black, red. So I usually use blue, white and red because it is more visible. Yeah, like this. And if you see there are too much white color, so then you can use that normalized gene expression divided by ST. So when you will click on this, you will see the the color have been improved, but now the normalized gene expression. So in this way, you can like the previously by this, you can download the Excel file, and using this you can download high resolution figure, which will open in this Adobe Illustrator, which you can later on save in the form of 300 or 600 dpi photos. So while moving to K-means analysis, so K-means analysis will divide all of these samples into different groups. So according to gene ontologies, how many number of groups can be divided using K-means. So if we will use this K-means, we can see that there are actually 4 clusters in the form cluster A, B, C and D. So cluster A were related to cell wall biogenesis, and here blue and red color shows down and up regulated genes. So you can also download this K-means data and high resolution figure. So similarly, we can see that this K-means analysis is according to the Go biological processes. So similarly, you can choose for cellular functions like this, and like we can see the overall details have been changed. You can also download in the form of Excel and Go molecular functions. You can also do this. So all the way you can also choose all the available gene data sets. So it will change using biological process, cellular component, molecular function. They will integrate all. So you can also download this high resolution figure. So the next important step from where the formally RNA-seq data analysis begin is the PCA, that is principal component analysis. So we can see that among the 6 samples, this shows the wild type, and this color shows the this color shows the mutant type, and this color shows the wild type. So this is the principal component analysis. You can also change the coordinate x-axis if you want this data into this and this. So you can also change, and you can also download high resolution figure. So DEGs 1, that is the differential expression of genes. Usually we use DE-seq 2 method to find the differentially expressed gene. So usually what we do, the FDR cutoff is normally used as 0.05, and minimum FC value should be 2 to get FC value. So I can just change this. So here we can see the wild type versus mutant type. So in this, it has actually taken the wild type as mutant type as the control and counting the in wild type that there are many upregulated. But in our case, we want to use wild type as control. So what we will do, so we can change the factor according to this. So we like if you want to change these, and like wild type, you want to change wild type versus mutant type, you can just simply resubmit. So because I want to use this, I want to know the values of genes were up and down in mutant type, and I want to take the wild type as control. So I will submit and recalculate. So when I will submit and recalculate, it will show you the number of genes regulated up and down regulated in mutant type and will take the wild type as control. So we can see that now it has been reversed, that it has changed to mutant type versus wild type. So you can also download this data in the form of Venn diagram. So you can simply you can split all the genes. You can click on this. Well, yeah, we can see their upregulated genes in mutant type were 784 and down-regulated genes were 393. So the totally number of 1,947 genes were totally up and down regulated. So you can also download this files in the form of Excel. So if I in the previous video I have briefly given all the detail. So here I am just going a quick overview. So here is the DEG level 2. We can also download volcano map, MA plot, and the scatter plot and high resolution figure. So we can see that this here is the enrichment trees and enrichment details and enrichment using string. So this function is added newly. So actually in this way, like this is the enrichment trees. Yeah, we can see that the number of gene ontologies which are down regulated and up regulated are mentioned. This is a beautiful feature which have been updated in the revised version, and you can also check this feature for cellular components. So it will show you the which cellular components were down and up regulated, and you can download and open in the form of audioble illustrator. So all of this all gene data sets can be simply integrated in the form of network, which is a new feature in this. So we can see here both up and down regulated genes. It will show, yeah, this is a useful feature by which you can understand your what processes and which pathways were up and down regulating in your mutant. So the photosystem 2 were down regulated and up regulated were related to these molecular mechanism and cellular components and biological processes were actually up and down regulated. So if you want to show only up regulated, you can simply click on this, and if you want only down regulated, so you can click on this. So you can also change the layout according to your end, and you can simply drag on this and make it visible according to your own choice. So I think so this is a wise feature added by their programmers to have a quick look on the pathways and genes. So here the next pathway is a pathway tab. So we can see that, so it will make a pathway tab of all. So here it is for all the processes. You can select like which process, like BP, CC, or MF. So which process you want. So if you want for all gene data sets, you can simply click on this. So we usually use the FDR value of 0.05. Yeah, 0.05, and number of top genes regulating, and that we can also change, but here mostly we choose number of pathways 30. So here we can also choose the different method, because here if this figure you think it is too long and difficult to use, so I will suggest to you to use this method, that is PGSE with all samples. So when you will click on this, it will automatically generate a heat map of all the significantly related genes of that pathway. So let's suppose from here you have chosen all the available gene data sets. So it will make top 30 pathways. It will integrate in the form of heat map. So that can be used in your submissions for your paper. Yeah, here is for all the pathways. So if we change it into the go biological process, so it will change into a new pathway by using this R integrated software. Sometime it is usually take, yeah, it we can see that it shows that there are no significant pathway found because this criteria we have used uh, is too strict, so we can simply relax it, and by just using pathway FDR zero point 0.1, because in some of the studies where FDR value 0.05 is not giving any significant pathway, so we can even move it to 0.1. But in some of studies, and in fact most of the studies use 0.05, but you can also choose zero point 0.1. So we can see that this in wild type and mutant type, number of genes DEGs that were related to this were down regulated in mutant type and up regulated in wild type. So you can simply just download this high resolution figure and you can use according to your own need. So this is the biological processes. So similar you can make for cellular components. Yeah, at this level there is no cellular component. So let's check for the molecular function. Yeah, so we can see that these molecular functions heat map. And if you want any specific one from these, like these are all molecular function pathways. So any of them, like let's suppose you want for kinase binding or ubiquitin. So from simply you can select here like the kinase binding. Yeah, we can see that. So it will specifically among all these genes. So it will make a heat map for kinase binding. So this feature is very useful in making the heat map and expressing your interest for your RNA-seq analysis. So next, it will all integrate all the DEGs into the genome. You can also select the comparison method FDR values and FC values. So it's okay. It is just asking there is an error. No problem. Yeah, we can see that here an error has been occurred. So check your logs or contact the app. So you don't need to just worry. You just need to change this FDR cutoff value to again to 0.5. So if you will run this PRDA program, so it may take few seconds. Although it is written that 5 minutes, but it usually don't take 5 minutes. So we can see that all the 12 chromosomes have been integrated for up and down regulated DEGs. So you can also select down regulated genes. So it will download the differentially expressed genes on all the chromosomes. So similarly, you can go for bicluster analysis. So if you will select the bicluster analysis, so we can say that if we make cluster at this values of including, so totally they found 11 clusters, and among WGC analysis we usually know that we use WGC analysis, and there are total 11 clusters, and cluster 1 has maximum genes, that is 470 genes. So we can also select here all the genes data sets. So among them, the maximum number of enriched genes were into these clusters and with their gene IDs. So you can also download this. And if you need to make a network of WGC analysis, so you can see here, we can see that total number of 11 modules have been found. But usually, as we know that this is actually soft threshold of 5. So as I can see that the number of modules are 11 are too high. So you can just simply change this soft threshold 5 into maybe 1. So you will see that the overall number of modules would be start to reduce. Yeah, now we can see that there are totally 1, 2, 3, and 4. So with the soft threshold of 1 and minimum module size should be 20. So now you see the total there are 4 modules in WGC analysis. So one is the turquoise, that is 490 genes. That is blue, here is 290 genes, and brown, that is 111 genes, and yellow, that is 102 genes. So you can also download this form, and you can also change our network layout for the interaction, which you can also use. And you can also download the heat map of all these clusters. So let's try to click on this. So it will create a heat map of 4 modules. We can see that this is turquoise, blue, brown, and yellow. All the genes uh, have been changed into cluster on the basis of WGC analysis. So that's all. If you still have any question to understand iDEP, or if you still face any difficulty in understanding or uploading the data, please let me know through my email, or please tell me in comments. Thank you for watching. Bye bye.